and gentlemen, thank you very much for asking me to speak to you today. My name is Tom Jefferson. I'm a physician. I'm an epidemiologist. I'm a co-author of 17 Cochrane reviews, mainly on vaccines. I'm here to talk to you about our work on HPV vaccines, human papillomavirus vaccines. I have some questions which I have agreed with Giovanni Guevara, and I have some answers. I will read the questions and then discuss the answers. The questions are numbered 1 to 5, and the references to our work, so the bibliographical references to our work, are in the document which you should have received. Please do not quote this document or any parts of it which are not in the public domain without my authorization. Thank you. So here we go. First of all, can you describe your work on HPV vaccines? The human papillomavirus vaccines are global interventions given to healthy individuals to prevent HPV-related diseases, such as cervical cancer. The vaccines are considered, quote, safe and effective, unquote, by healthcare authorities. Benefits of the major outcomes, such as cancer and deaths, will only be verifiable after decades. But safety signals raise concern whether selective presentation of the evidence had influenced the authorities' considerations of benefits and harms. Now, you should have read, as the first line indicates in the document, you should have read our little introduction called Redefining the E in EBM. Now, this is very important that you read this, so you will be given the background to what I'm about to say. So if you haven't read it yet, do so now as you get to the document. The link is on top of the document. Because of the widely perceived threat by reporting bias to the validity of trial journal publications, that's reporting bias means selective bias. I choose what to report. Perhaps the most famous form of this is publication bias. But because of the threat posed by reporting bias of all kinds, we investigated the benefits and harms of the HPV vaccines by going directly to regulatory submissions made to different regulators, and I will list them as we go along. And we did this, first of all, by building an index of the study programs. So we accessed clinical study reports from the HPV vaccine study programs and also compared these reports to corresponding study documents like trial register entries and journal publications. And this basic preliminary part of our work is published, and you will have the reference in the document under 1. The study programs, the reconstructed study programs of the HPV vaccine trials demonstrated deficiencies and variabilities in the availability of study data in trial register entries and journal publications. This is exactly what we found with Tamiflu a decade ago. So that journal publications are a very dangerous source of data and information. And trial register entries are sometimes not updated, and they contain mistakes. For example, only half of the completed studies of HPV vaccines listed on clinicaltrials.gov had results posted. A third of all studies were not published. That's why we constructed an index. If you like, it's like a database of all studies, and we listed 206 prospective comparative studies sponsored both by pharmaceutical and non-pharmaceutical agencies. We then accessed regulatory data on HPV vaccines from the European regulator, European Medicines Agency. Our analysis reporting accessing the HPV vaccine trial data from clinical study reports showed that regulatory policies are in need of change to increase transparency and fluency and usability. And our results are published in the BMJ, were published in the BMJ this summer. And again, the reference is in the document. To give you an example of the problems that we had, it was not possible to get a complete set of unredacted clinical study reports on the HPV vaccines. So we have 24 reports, but they're all missing some bits. Even so, we're looking at 64,000 pages. Compare that with 10, 15 pages on average of a trial publication. The next phase, number 3, was minimally biased evidence synthesis. What does that mean? Our systematic review of 24 clinical study reports with nearly 100,000 participants. This is undergoing peer review at the moment, but I can tell you that the review showed that at 4 years follow-up, HPV vaccines decreased HPV-related precursors to cancer, and they decreased also treatment procedures for gynecological and mainly cervical reasons. But the vaccines increased serious nervous system disorders and general harms. The trials used biased designs and underreported harms, which prevented adequate harm assessment. Now, as I said, this is under consideration for publication, this work, but you can find the protocol and its amendment on Prospero, and the link is in the document. Next came the comparisons of published trials with the clinical study report versions. Our comparison of corresponding study documents showed no effect differences or pooled estimates. Okay, great. So we can use publications. It doesn't make any difference. But we demonstrated that the clinical study reports were quantitatively and qualitatively superior with more outcome data and information that improved risk of bias judgments. You've got to bear in mind that one possible explanation for the finding of no difference between clinical study reports and publication is that distortions in the trial designs ensures that the same results are reached despite the data source used. That means that whether you use publications of 15 pages or clinical study reports of 10,000 pages, if the trial has a biased design and the reporting is biased, in general, reporting of, for instance, of harms is fragmented, it's in very many different categories. Some of them have got definitions and some of them have not. And the actual surveillance is fragmented and cannot actually be compared from one trial to the next. Well, whether you use a clinical study report or a journal publication as a source of data doesn't actually make much difference. What does make a difference is that you can see these distortions in a clinical study report and rarely in a publication. The second question is, what are the main problems with the evidence of efficacy and safety of the HPV vaccines? It is not clear to what extent the HPV vaccines' benefits outweigh their harms, as the study programs and clinical study reports were influenced by reporting bias and biased trial designs. The clinical study reports were superior study documents that contain better information for risk of bias judgments. Clinical study reports should therefore be used for systematic reviews of important interventions, such as HPV vaccines. The next question, number 3 that I was asked, was what are the main problems with the Cochrane HPV vaccine review, published in May 2018 by Arbin et al. Well, I've given you I have put a link to our main criticisms, which are published on BMJ Evidence-Based Medicine, BMJ EBM. They're listed there and you can read them. But just briefly, the reviewers, the Cochrane reviewers, ignored evidence of bias, despite being sent our HPV study index 4 months before publication. The Cochrane authors based their review on journal articles and made some very fundamental mistakes, such as referring to active comparators as placebo and misclassifying 4 deaths. They attribute to them the deaths to a control arm, when in fact it was the intervention arm. What do we mean by active comparators described as placebos? All of the trials, except for 2 Merck trials, a Gardasil and the Gardasil 9 trials, have comparators which there are either their adjuvants, the adjuvants of the vaccine, or another vaccine, like hepatitis A and hepatitis B. And these comparators are active. There's nothing more active in vaccinology than an adjuvant. There's nothing more active in vaccinology than another vaccine. Despite this, the authors describe them as placebos, and as placebos, they are described in the majority of publications. Which brings me back to my original point about accessing clinical study reports. We know exactly what were in those placebos. We know each dose of those placebos, so-called placebos, and I can assure you that were not placebos, with 2 exceptions. One is a Gardasil trial, which was requested, in fact, specifically by FDA. So somebody at FDA must have been pretty nervous about these, the safety of these vaccines. But essentially, the so-called placebo was normal saline, they described it, but in fact it wasn't. It had a carrier solution. It was the carrier solution which had some excipients, things like Twin 80 and povidone, and we don't know what these substances, what the effects of these substances are. The other smaller trial, Gardasil 9, was a placebo-controlled trial, but the problem was that the participants had been pre-exposed to Gardasil 4. So they'd all been exposed to at least 4 different virus-like particles and the adjuvant. So the placebo was actually given to people who had already had 1 or more doses of Gardasil. The next question is, what are the current challenges in the paradigm of evidence-based medicine? And these are described, as we can see them, they're described in the Redefining the E in EBM publication summary, which I mentioned and which is linked again at the beginning and at question 4 of this document. Briefly, as you will have gathered by now, the main challenge facing us is the use of more reliable and detailed sources of evidence than journal publications, and probably also more detailed than register entries. Remember, register entries are not updated and they do not contain methods. They're also unstructured in IMRAD, Introduction, Methods, Results and Discussion form. So they're not easy to consult. Clinical study reports are extensive documents which run into thousands of pages, but they are structured along similar lines to IMRAD. So they're familiar. And if you don't want to read the whole clinical study report, you can read a synopsis, which is usually anything between 5 and 15 pages, which packs a lot more information than any publication. The bias is also minimal because although these are still commercial publications, clinical study reports are written to get vaccines registered, the same as publications are written to have vaccines sold, vaccines or pharmaceuticals sold. Despite the fact that they're still commercial publications, they have a considerable amount of detail that allows you to go in and do this kind of research work that you need to do to understand what the evidence and what the intervention are. Okay, the last question that Dr. Guevara posed to me was, how are you following up your HPV vaccines work? Well, some of you may be aware that one of us, my colleague Peter Doshi, won a court case in the summer against Health Canada in the federal court, in the Canadian federal court. Health Canada is the Canadian regulator. The judge ordered that thousands of files, regulatory files, were released to Peter Doshi and the two other named claimants, that's myself and a statistician. That means that we will receive, well, Peter Doshi's already received, we're waiting for IRB clearance. We received thousands more regulatory files from Health Canada as a consequence of this decision. Essentially, we have as much as Health Canada had. So probably everything that a regulator should know and not necessarily use. So further analysis and integration of what I have just said to you, of the available reviews, an update of available reviews, an integration of available reviews is possible with an unprecedented level of detail, down to individual participant data and down to individual case report forms. That's the notes that are kept on each participant of each trial. And that, ladies and gentlemen, concludes what I have to say to you. And again, thank you very much for giving me the chance of speaking to you. Finito.